Bismillahirrahmanirrahim. Welcome to the second part of dietary lipid metabolism. So, free fatty acids, free cholesterol, and 2-MAG are the primary products of lipid digestion in the jejunum. These plus the bile salts and the fat-soluble vitamins A, D, E, and K form mixed micelles. The mixed micelles are soluble in aqueous environment of the intestinal lumen. These particles approach the primary site of lipid absorption, the brush border membrane of the enterocytes. The hydrophilic surface of the micelles facilitates the transport of the hydrophobic lipids through the unstirred water layer to the brush border membrane where they are absorbed. Bile salts are absorbed in the terminal ileum with less than 5% being lost in the feces. Relative to other dietary lipids, cholesterol is only poorly absorbed by the enterocytes. Drug therapy, for example with ezetimibe, can further reduce cholesterol absorption in the small intestine. Finally, because short and medium-chain fatty acids are water-soluble, they do not require the assistance of mixed micelles for absorption by the intestinal mucosa. Alright, so after absorption by enterocytes, we will talk about the triacylglycerol and CholesterylEster resynthesis. The mixture of lipids absorbed by the enterocytes migrates to the smooth endoplasmic reticulum where biosynthesis of the complex lipids takes place. The long-chain fatty acids are first converted into their activated form by fatty acyl-CoA synthetase, which is also known as thiokinase. Using the fatty acyl-CoA derivatives, the 2-MAG absorbed by the enterocytes are converted to TAG through sequential reacylations by 2 acyltransferases. The first one is acyl-CoA monoacylglycerol acyltransferase, and the second is acyl-CoA diacylglycerol acyltransferase. Then lysophospholipids are reacylated to form phospholipids by a family of acyltransferases, and cholesterol is acylated primarily by acyl-CoA cholesterol acyltransferase. Virtually all long-chain fatty acids entering the enterocytes are used in this fashion to form TAG phospholipids and cholesterol esters. Short and medium-chain fatty acids are not converted to their CoA derivatives and are not reesterified to 2-MAG. Instead, they're released into the portal circulation where they're carried by serum albumin to the liver. Next, we'll talk about lipid malabsorption. Lipid malabsorption resulting in increased lipids in the feces, a condition known as steatorrhea, can be caused by disturbances in lipid digestion or absorption. Such disturbances can result from several conditions including cystic fibrosis, causing poor digestion, and short bowel syndrome, causing decreased absorption. The ability of short and medium-chain fatty acids to be taken up by the enterocytes without the aid of mixed micelles has made them important in medical nutrition therapy for individuals with malabsorption disorders. Now we'll talk about the secretion from enterocytes. The newly resynthesized triacylglycerols and cholesterol esters are very hydrophobic and aggregate in an aqueous environment. Therefore, they must be packaged as particles of lipid droplets surrounded by a thin layer composed of phospholipids, non-esterified cholesterol, and a molecule of protein, ApoB48. This layer stabilizes the particle and increases its solubility, thereby preventing multiple particles from coalescing. Microsomal triglyceride transfer protein is essential for the assembly of all tag-rich ApoB-containing particles from the endoplasmic reticulum. The lipoprotein particles are released by exocytosis from the enterocytes into the lacteoles. The presence of these particles in the lymph after a lipid-rich meal gives it a milky appearance. This lymph is called chyle, and the particles are named as chylomicrons. Chylomicrons follow the lymphatic system to the thoracic duct and are then conveyed to the left subclavian vein where they enter the blood. Once released into the blood, the nascent chylomicrons pick up apolipoprotein E and C2 from high-density lipoproteins and mature. Now we'll talk about the use by the tissues of these products. Most of the triacylglycerols contained in chylomicrons is broken down in the capillary beds of skeletal and cardiac muscles and adipose tissue. The triacylglycerol is degraded to free fatty acids and glycerol by the lipoprotein lipase. This enzyme is synthesized and secreted primarily by adipocytes and muscle cells. Secreted LPL is anchored to the luminal surface of the endothelial cells in the capillaries of muscle and adipose tissues. Familial chylomicronemia, which is also known as hyperlipoproteinemia, is a rare autosomal recessive disorder which is caused by deficiency of LPL or its coenzyme ApoC2. The result is fasting chylomicronemia and severe hypertriacylglycerolemia, which can also lead to pancreatitis. Now we'll talk about the fate of free fatty acids, the fate of glycerol, and the fate of chylomicron remnants one by one. Now the free fatty acids derived from the hydrolysis of triacylglycerols may either directly enter the adjacent muscle cells or adipocytes or be transported in the blood in association with serum albumin until they are taken up by the cells. Most cells can oxidize fatty acids to produce energy. Adipocytes can also reesterify free fatty acids to produce triacylglycerol molecules, which are stored until the fatty acids are needed by the body. And finally, free fatty acids are transported in the blood until they are taken up by the cells. Next, we will talk about the fate of glycerol. Glycerol that is released from triacylglycerols is taken up from the blood and phosphorylated by hepatic glycerol kinase to produce glycerol 3-phosphate, which can enter either glycolysis or gluconeogenesis. After most of the triacylglycerol molecules have been removed, the chylomicron remnants, which contain cholesterol esters, phospholipids, apolipoproteins, fat-soluble vitamins, and a small amount of TAG, bind to receptors on the liver, and then they are endocytosed. The intracellular remnants are hydrolyzed to their component parts. Cholesterol and the nitrogenous basis for phospholipids, for example choline, can be recycled by the body. If removal of remnants by the liver is decreased because of impaired binding to the receptor, they accumulate in the plasma. This is seen in the rare type 3 hyperlipoproteinemia, which is also called familial dys beta lipoproteinemia or broad beta disease.